All right. Now we are going to talk about translation. We are no longer going from Richmond to jolly old England, where we are going to speak the same language with a little different accent. We're going to go to a completely different language now. We are going from Richmond to Japan, and we are going to have a completely different language that we have got to master. Okay? So, having said that, let me get to my, okay. The language that we are going to shift to is the language of amino acids and proteins. So we've got to get from this nucleic acid language to the protein language. Okay, don't forget our central dogma. Okay, from DNA to RNA through transcription. Now we've got to take this RNA molecule out of the nucleus where it's made, because that's where transcription occurs, because remember, DNA can never leave the nucleus, so we're going to have the master DNA being copied in the nucleus. Now we're going to take this molecule out of the nucleus and into the cytoplasm where protein will be made from it. Okay? That process of making protein from this RNA instruction template is called translation. Okay? And then of course the proteins are what give us our traits. Okay? So, please remember the central nature of the ribosomes. They are where the proteins are being made. Ribosomes exist out in the cytoplasm. So again, if this is my cell, this is my nucleus, here's this ER stuff, and there are ribosomes that sit on the ER, and while they are sitting there, they make proteins. That's the rough ER, if you'll recall from your first year biology. There may also be ribosomes just floating in the cytoplasm and making their proteins. Ribosomes are two-part structures. There's a big subunit and a small subunit, and this is a kind of an accurate depiction of kind of the shapes of ribosomes, if you will. Okay, so this is where the RNA instructions will be read and the protein will be built according to those instructions. Okay? So, the complete mRNA molecule is going to have to leave the nucleus and go to the ribosome. It's been processed, the introns have been cut out, the caps and the tails been added, and so this finished RNA, messenger RNA, that is shown right there, is going to come out into the cytoplasm, where the two ribosomal subunits, the big subunit and the small subunit, will kind of sandwich it like two pieces of bread. Now, that RNA is going to tell us how to build the protein, and what that means is it's going to tell us of those, if you'll remember, there's 20 different amino acids. It's going to tell us which one goes first, which one goes second, which one goes third, fourth, fifth, sixth, and so on. And that order of amino acids is going to be really important for determining how the shape that the protein takes. But how do we know what amino acid goes where from looking at a molecule of RNA? How can that tell us anything? Well, it can tell us because we have figured out the genetic code. It is based on the order of nucleotides in that RNA, which of course is based on what was in the DNA in the first place. And the genetic code looks like this. You might recall seeing a chart like this from your freshman year. Please note, the same code is used for all life on Earth, suggesting a very fundamental relationship between all living things on planet Earth. Okay? Common origin of life is suggested, given that all of life uses the same genetic instruction sheet. Okay? Now, the mRNA molecule is going to carry that genetic message away from the DNA and to the ribosome in the form of little tiny words, little 3 nucleotide words, quote unquote, called codons. You need to know this term, codon. Codons are what we are seeing in this genetic code chart. Okay? Now, whoops, let me go back. There are some special codons, and those have been highlighted in red. The rest of the codons, if you look here, are coding for a particular amino acid. For example, if you are looking at an RNA molecule and you see, well, let me go over here. You see an adenine, a cytosine, and a uracil, okay, right next to each other. That's going to code for this particular amino acid, and I have no idea what THR stands for, so I shouldn't have picked that one. How about this one? Cytosine, cytosine, cytosine next to each other in the RNA molecule. That is telling me we need I believe a proline amino acid. Okay, so 3 nucleotides, 3 RNA nucleotides on that mRNA strand are required to tell us what amino acid we need to put in to a certain spot. It's a triplet code. Okay, a series of these codons are 3 nucleotide words, so to speak. Okay, that's a nucleotide, that's a nucleotide, that's a nucleotide. You see those 3 nucleotides next to each other in the mRNA strand, you know we need phenylalanine amino acid. Okay, there are 64 code words altogether in this code, and each triplet is called a codon. We've got all that. All right, each different codon codes for a different amino acid, and they're read in the 5 to 3' direction. We got that. All right, now let's talk about these special codons that were highlighted. The start codon, the ribosome. Here is the ribosome, big and small subunits. Here is the mRNA being woven through, and these are the little nucleotides exposed on the RNA. This ribosome might clamp on anywhere. It needs to know where is the start. So it is going to look for a particular 3-letter, 3 nucleotide sequence to tell it when to start. And that we would recognize right here as AUG. Okay? AUG is always the start codon, and it always codes for one particular amino acid called methionine. We'll just call it MET. Okay? So, MET then is always the first amino acid in any polypeptide because that's the start codon, the start amino acid coded for by the start codon. Often it is removed as part of the processing of the protein after it's made. Usually that gets removed, but it's always the first one placed there. The ribosome also needs to know when to stop reading this RNA transcript, and so there are 3 stop codons, UAA, UAG, and UGA. And they don't code for anything, they just tell that ribosome when to let go of the RNA transcript. Alright, now we have one more player we need to talk about, and that's tRNA, and the T stands for transfer. This molecule of RNA transfers something from one place to another. Okay? The T stands for transfer, and what the tRNA is transferring is it is bringing amino acids from the cytoplasm to specifically to the ribosome. Okay? There's lots of tRNA molecules floating around in the cytoplasm. Let's draw one. I think I will draw my tRNA today looking like this. Okay? And it will have 3 nucleotides. Oh, I don't know. Maybe it's got a U, a C, and a C right there. Those represent 3 nucleotides. And here it's carrying an amino acid, a particular amino acid. And what it's going to do, it's going to float up to the mRNA that is in this ribosome and try to see if it's complementary or not. And that's how we'll know if this amino acid is needed to build the protein we're trying to build. So each tRNA molecule, of which there are lots and lots of them floating around, each different type of tRNA molecule is carrying around one of the 20 different amino acids that exist. Okay? And a cell also has to have lots of amino acids in it all the time because it's always making protein. Cells can achieve this either by making amino acid themselves from building blocks or simply taking them up from food and reusing them, which we do quite a bit. So, if we were going to look at a more realistic depiction of a tRNA molecule, its shape, the actual RNA folds crazily on itself, looking like that. The overall shape, you can see how they've tried to make an overall simpler geometric shape out of it here, but in reality it's this ribbony thing. Now here they've kind of like ironed out and flattened it so we can see, and we don't really care about all of this RNA all through here. What we care about is this RNA, because when I drew that with my amino acid, these three UCG or whatever, that's what's down here. This is called the anticodon, and we'll talk about that in a minute. Okay, the anticodon is on the middle of the three-leaf clover here. Okay, now there's three different ways to look at it. It's here, it's here, it's here, but these are all just tRNA molecules drawn in different ways. This here, this end right there, is where we're going to attach the amino acid that the tRNA molecule is going to carry right there. It's just depicted in three different ways. So the amino acid is attached to the 3' end of this. You don't need to remember that, but it is attached to that specific end. Now, the anticodon, okay, is a nucleotide sequence that is complementary to a codon that is on mRNA. Remember, it is the mRNA codon that is in the genetic code chart. So please don't start trying to decode anticodons. They are not what's in the code chart that we saw a few slides ago. Nonetheless, the cell needs to be able to deal with these anticodons and match them to codons to know if this particular tRNA molecule has the right amino acid that we need. Okay, let's look at it. If the mRNA codon, so here is my ribosome, here is my mRNA, and it is looking at UUU. That's the codon I'm looking at, okay? Well, I would look at my genetic code chart and see whatever UUU coded for, and that would tell me as a human being on the outside what's going on. But the way the cell deals with this is that there are tRNA molecules, and they are all carrying different amino acids, and I want to know that if this tRNA is bringing the correct amino acid up, it will have the complementary sequence to UUU, which would be A. And if this is correct, then it will leave this amino acid behind with the ribosome. So the ribosome can use it to make the protein that we want. And remember, it's the proteins that make up our bodies. Okay? So the tRNA molecule with the AAA codon, in this anticodon rather, in this case, will always be carrying the amino acid, there it is, Phe, which is phenylalanine. So if UUU is seen on the mRNA, then we must need the amino acid Phe, and then a tRNA molecule with the complementary anticodon will come up carrying Phe and drop it off. And that's how we get the correct codon, the correct amino acid where we need it. Okay? So, as the genetic message is translated, again, here's my ribosome, here's my mRNA, boom, 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 and it's going to just read it along. It'll see, for example, UUU, and so we bring up the Phe amino acid, then we shift to the next three. What do we need? What's next? Maybe it's CCC, I don't know. And so a tRNA would come up complementary to that and leave its amino acid, and so on. So what this diagram is showing, here's our ribosome big and small subunits, there's the mRNA molecule here, This is the tRNA molecule here with its anticodon, seeing if it matches, and we see here the growing polypeptide strand. I think I have a better picture coming up. All right. Now, understand that a tRNA molecule, okay, so let's say this guy brings his amino acid, whatever it is, up, and it's the one we need at the ribosome, and so we're going to drop it off. Well, then this guy is empty. He doesn't have one, and we've got to load him back up again and recycle him. So there is an enzyme that does that. It's called aminoacyl tRNA synthetase. Synthesize means to make, ACE means it's an enzyme, and we're going to make tRNA carrying an amino acid. Okay? So it's basically an enzyme that's going to bond an amino acid to a tRNA molecule. It requires a little bit of energy, so we're going to need to spend some ATP for this. The bond is not terribly stable. When we attach, look, here is the enzyme, an enzyme that is going to attach this amino acid to this tRNA molecule right here. It's not a really stable bond. Why is that important? Because ultimately we want to release this amino acid from the tRNA and leave it with the ribosome to build a protein. So it makes sense that it's not a very stable bond that's created when we attach this to the tRNA molecule. It's kind of unstable, which means this will easily come off and help us build the protein. Okay? So, I don't think we need all this detail. I do think you should be aware that since there's 20 different amino acids, there's 20 different versions of the enzyme to add an amino acid to a tRNA molecule. There's one version for each of the different amino acids. And you're just seeing the active site here. We're seeing a tRNA molecule come up. We're seeing the appropriate amino acid come up. They get fused together. And then we have the tRNA molecule carrying the appropriate amino acid ready to go do its job. Okay? Okay, that's all we need to know about that. Alright, so understanding, remember what we said, that ribosomes are made of RNA and protein. Is it an organelle? Is it an enzyme? It's causing a molecule to get built. It seems like it's kind of got an enzyme property about it. You can debate that. It is pretty big for an enzyme. It's those two subunits, the big and the small. We are going to focus on the big subunit because the big subunit has got 3 regions in it that you can see labeled there that we need to learn about. So let's talk about those. So the ribosome has something called an A site. We're going to call this the active site. And it is going to be, well, let me see if we can kind of not, let me spell out everything yet. Let me just give you the other site. Another site is the P site, and we're going to call this the passive site. And this is kind of, well, exactly as it suggests, passive. We'll talk about what this means. Okay, and then we've got the E site, which is exit. I think of it as exit. Okay, so A, P, and E, and these are being shown right here. Here's the RNA strand. Here is a tRNA molecule with its amino acid, and this is the A, the P, and the E. Okay, regions on the ribosome. Now, okay, just to remind you of some stuff we talked about before, a ribozyme or a is kind of an RNA molecule acting as an enzyme. In this case, the ribosome is kind of like a huge RNA molecule. The protein that is in it is more there to stabilize the whole thing. The RNA is really acting as the catalyst to make everything happen. Just reminding you that RNA can act as a catalyst. Let's see how it works. Again, much like what we saw with transcription, we're going to have initiation, we're going to have elongation, and we're going to have termination. We're going to start it, we're going to do it, and then we're going to end it. Initiation is going to bring together the things we need to do the translating, the making of the protein. We need the mRNA because that's got the instructions from the DNA. We need the ribosome because that's what's going to do this for us. And we need the initiator, the first tRNA. And what that means is we need the tRNA that is carrying the methionine amino acid, because remember, that's always the first one. Okay, so elongation, we're going to be just adding the amino acids, and then we're going to end it. So let's talk about initiation. Let's bring together the mRNA, there it is in the red, the two ribosomal subunits, the big and the small, there they are, okay, and the tRNA we need is the one that is carrying the methionine amino acid. So that's coming over here, okay. Now, that tRNA has got the very first amino acid in the that is going to make up the polypeptide we want to make, whatever it is. Maybe it's the polypeptide that makes pigment in your skin or whatever. Okay? Whatever it is, the first amino acid in it will always be methionine. All right? Now, this is what it looks like when we get started. That initial tRNA molecule carrying MET is going to start out in the P site. This is the only time that ever happens. Normally the tRNA is going to come into the A site, but this initial one comes into the passive site, and it finds this anticodon that is on the tRNA. The anticodon is going to complement the start codon that's on the RNA here in red. Okay? And so now we're all initiated, we're ready to start reading and building our protein. So elongation has got three parts. All right, let's look at the three parts. Codon recognition. Okay, we are going to try to recognize when the codon of the incoming tRNA bases with the complementary mRNA sequence. Okay, so that is we are looking at right here. This is our initiation complex. Then we've got the next tRNA molecule that's going to come in, and hopefully it's going to be complementary to this codon here on the mRNA. If it's not, it won't be able to bind because it's not complementary, and then it'll go away. If it is, it will come in here and it will stay for a little while. Okay, so that's codon recognition. Now, peptide bond formation. Remember, peptide bonds are the bonds between amino acids. If this is an amino acid, amino acid, amino acid, these are peptide covalent bonds between them. We got to build a polypeptide, so we need to create the bonds between the amino acids. Okay, so let's look at the picture. So now this that was coming in, this tRNA, is now docked here in the active site. So what happens is, in the active site, this whatever is on the tRNA at the P site is going to bind a covalent bond to the amino acid that is on the A site. So these amino acids will bind to this one, so that, if you look down in the next slot, this tRNA molecule is empty, and this is now carrying all the amino acids, this one that's in the A site, and this P site one, passive site, is empty. Okay, so we formed a peptide or covalent bond between these amino acids, and as you can see, the whole chain got one amino acid longer right there. Okay, now we need to shift over to read the next section of mRNA. This is called translocation. We're going to move what was in the A site now to the P site, the passive site, where it will wait. Okay, what was at the passive site is now going to move to the exit site and leave. So let's look. So now we are going to shift everything over. we are going to shift down to the next codon to be read, which is this one. So this is now going to move to the exit, and you can see it's going to leave and hopefully go get a new amino acid. This that is here at the A site is shifting now to the P site, and now we can read the next codon. Okay, and repeat the process all over again. Okay, so translocation, basically everything shifts over by one codon. Now, termination is when we hit a stop codon. If you remember, we've got 3 possibilities of stop codons, and if we hit one, something called a release factor, a protein, will attach and cause the whole thing to just release. Everything will let go, and the polypeptide will be able to go off and do its job, whatever that would be. So let's look. Actually, no, I lied. There is no looking at that. If we hit a stop codon, release factor is going to bind, and it's simply going to disassemble the whole ribosome and the mRNA and the protein that was made and everything else. Okay? Alright, this idea of polyribosomes is simply multiple ribosomes translating one piece of mRNA, and you can see this actually happening in this electron micrograph. All these ribosomes are translating this one mRNA strand, and so that's how a lot of protein can get made at once. Not to mention there can be multiple mRNA strand copies being translated all at the same time in the cell. Now, when a polypeptide is made, usually it's not perfect. It needs to be modified to actually make it work. Normally this involves folding, because if you'll remember, amino acids, there's a central carbon, there's a carboxyl group on one side, an amino group on the other side, there's a hydrogen, and then there's that funky R group. You remember that? And those R groups will interact with each other, and the whole thing will fold very complexly and get a particular shape. Okay? So let's kind of review real quick. You remember primary structure, secondary structure, tertiary structure. That's the order of the amino acids. Then the secondary structure is the hydrogen bonds that cause it to make a beta pleated sheet or an alpha helix. If you don't remember any of this, you might want to go back and review a little bit. Now, there are chaperone proteins that will help this molecule kind of fold the way it's supposed to fold. And sometimes amino acids are actually modified or removed. For example, that MET amino acid, that's the first one, is almost always removed. So the polypeptide may be cut. 2 or more polypeptides may come together. Remember quaternary structure, when you bring together 2 or more polypeptides. Hemoglobin is actually 1, 2, 3, 4 polypeptides all together. We talked. There's a great picture. This is exactly the picture we looked at in class. Primary structure, secondary structure, tertiary, and then quaternary level of structure. Okay. This slide is enough to make you want to cry. There's so many words, but it is just helping you understand that there are many roles of RNA in cells. We've talked about several of them: mRNA, tRNA, rRNA, the primary RNA transcript, or the pre-RNA, which is mRNA, which is before we've processed it. Small nuclear RNA, which helps to make, makes the SNRPs, which make up spliceosomes. I wouldn't worry about these 3, but we have talked about all 5 of those. All right. This is reminding you why RNA is so much more versatile. It's really because it is single-stranded, and so these exposed bases can interact with things, whereas in DNA the bases are all tied up to each other and they can't interact. So it can hydrogen bond between itself, it folds on itself, it can actually add functional groups to itself and act as a catalyst. So it's kind of a very amazing molecule in the things that it can do. Okay, so let's just look at this example. Okay. There's your DNA. If this RNA comes from that, we get this protein that we would use our genetic code chart. We would see that for every T there would be an A, for every A U, C to G, and so on. So this is how we would transcribe this molecule. If we saw a T, we'd give it an A. If we saw a T, we'd have an A, and so on. And then once we have this, we just use our genetic code chart, and we'd say, all right, there's AUG. What's that on the genetic code chart? Well, that's MIT. CGU, what's that? Look it up on the chart. It's ARG, and all of that. Okay. There you are coding in triplets, and there's your codons. It gets a little bit off by the time. There's your codon for the last one. Okay, Francis Crick of Watson and Crick fame. He was one of the ones that helped determine the codon system. Let's see, I don't think we need any of that. I don't think we need that. And just remember, codes for all life. You should be aware that the code is redundant. Notice that there are several codons that code for phenylalanine. Look, all of these that code for serine. So there are several codons that code for each amino acid. It's usually the third base. If you look, UC, 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 all the same, and it doesn't really matter what the third base is here. They all code for serine. It's that third base that kind of seems to wobble around. And don't forget the stop codons and the start codons. Okay. I'm just trying to see. I think we might be done. That's our central dogma. This is a great slide here. Telling the story all the way from transcription to translation. Okay, this is the pre-mRNA strand. We have to process it though, cutting out the introns, 5' cap, poly A tail, all of that. It comes out as a mature RNA strand, and then it attaches at the ribosome, the active site, the passive site, the exit site. There's your tRNA molecules, your growing polypeptide strand right there. I think, this is showing you the enzyme that's making the connection here between the amino acid and the tRNA, so that we can make the tRNA molecules. There's the growing polypeptide. If you can understand that diagram, you understand a whole lot. We'll talk about it in class. Okay. All right. This is just yeah, I think we'll stop here. All right, thanks a lot for watching. I'll catch you on next time. Bye.